The great mystery of the origin of life. How did it come about? Maybe everything was as shown in fantasy films? Once upon a time, gray-blue giants flew to the planet and sowed the Earth with necessary molecules. Well, nature and time have already brought this matter to a logical conclusion. This scenario doesn't seem so fantastic against the backdrop of, for example, the achievements of modern biotechnologists. Meet the xenobot, a programmable organism made from living cells. You could say it's a biological robot. Under the microscope, you can see how in a liquid medium, small clumps in petri dishes move chaotically. They move around, self-organize, and even carry loads. But if you flip the clump over, it will just lie there like a turtle flipped onto its back. Their behavior reminds of microscopic flatworms or even tiny slowpokes, distinguished by a complex body structure despite their small size. But the similarity in behavior is deceptive, as the clumps consist of only two types of cells: skin cells and heart cells of frogs. First, they took stem cells from frog embryos and differentiated them into heart cells, which have the property to contract, and skin cells, which do not have this property. Then they combined these active and passive components, using the natural tendency of cells to stick to each other, encountering individual cells along their way. The clusters gather them into small heaps, and if you cut the xenobot in half, it will reconnect, just like the T-1000 from the movie Terminator 2. But maybe everything happened without any alien technologies, without a landing of little gray men, and life was brought to the third planet from the sun in a different way. After all, the theory of life originating on Earth from space is successfully developed by many scientists. For example, a solid international scientific team led by Australian immunologist Edward Steele, with the participation of astrobiologist Chandra Wickramasinghe, an honorary professor at Buckingham University, director of the Center for Astrobiology, published a review article in the journal where it analyzed the hypothesis that life on Earth originated from extraterrestrial sources. Starting from the 1980s, Wickramasinghe, a British physicist, astronomer, and astrobiologist, noted traces of organics on comets as clear signs of microbial life. The scientist believes that many genetic features could not have arisen through random mutations, and allows for the possibility that comets that hit Earth contained frozen biomaterials. The proof of their viability was an experiment by Swedish scientists studying tardigrades in open space. The samples were divided into three groups. One of them, upon arrival in orbit, was subjected to vacuum conditions and exposed to cosmic radiation. Another group was subjected to ultraviolet irradiation and from 280 to 400 nanometers. The third group of animals experienced the effect of the full spectrum of ultraviolet from 116 to 400 nanometers. All tardigrades were in a state of anabiosis. After 10 days spent in open space, practically all organisms were desiccated. But the tardigrades on board the spacecraft returned to normal. Most of the animals exposed to ultraviolet radiation with a wavelength of 284 nanometers survived and proved capable of reproduction. Only 12% of the animals from the third group survived. Nevertheless, the survivors were able to produce normal offspring, although their fertility was lower than that of the control group on Earth. This experiment proved that tardigrades can survive in space conditions. At the same time, the journal Geochemica et Cosmochemica Acta published the results of a study of dust particle samples taken from the surface of a comet. In these samples, scientists from the University of Arizona found minerals that can only form in the presence of liquid water. Here's another fact. Spectral analysis of the comet Gale showed the presence of organic molecules in it. If you add to this the already proven fact that comets can contain liquid water, it turns out that travel for microorganisms becomes not at all difficult. Moreover, as seen in the example of tardigrades, some organisms don't even need water to comfortably withstand cosmic temperatures and radiation. These tiny creatures, which appeared in the Cambrian period, pose a serious challenge to traditional thinking. Their space-resistant qualities are fully consistent with evolution in extraterrestrial conditions of space and are incompatible with purely terrestrial conditions. The same applies to modern and ancient ones from 500 million years ago. There are interesting data and a question that became one of the first memes of humanity. Is there life on Mars? Almost half a century ago, 2 Viking landers became the first American devices from Earth to land on Mars. They made the first high-quality images of the planet, studied its geographical features, and analyzed the geological composition of the atmosphere and surface. But most interestingly, they also conducted experiments in search of microbial life in Martian soil. Overall, these life detection experiments produced unexpected and contradictory results. One of the labeled release feller experiments showed that Martian soil gives a positive result for metabolism, but no traces of organic material were found. Where could metabolism come from if there was no organic matter? The protocol for this experiment was developed by Gilbert Levin. During it, both Viking 1 and Viking 2 collected samples of Martian soil. They introduced a drop of diluted nutrient solution into them, and then observed the air above the soil, expecting to see signs of metabolic byproducts. Since the nutrients were marked with radioactive carbon-14, if microorganisms in the soil metabolized the nutrients, they should have produced radioactive byproducts like radioactive carbon dioxide or methane. Before the experiment was launched, the experimental protocol was tested on the most diverse Earth soils in inhospitable places for life, from Death Valley to Antarctica. In any case, the experiments gave a positive result. They found life. Then, as a control, the scientists heated the samples to 160 degrees Celsius to kill all forms of life, and they got a predictable negative result. To confirm that the experimental procedure would not give a false positive result, the scientists tested it on sterile soil, which also gave a negative result. For decades, Gilbert Levin was not allowed to publish a scientific paper reviewing the results of that long-ago experiment, which was considered a failure, and only relatively recently was he able to do so. And not just because the scientific publication by Gilbert Levin and his colleague Patricia Strott was, in the author's words, so thoroughly licked that it is now impossible to attach oneself to it. The matter is also that in 2014, the Curiosity rover's laboratory first discovered organic molecules on Mars. The sample analysis at Mars laboratory found there, in particular, methane, chlorinated hydrocarbons, and other organic molecules. Another significant discovery was made by astrobiologists from Cardiff University. They analyzed fragments of a meteorite that fell in Sri Lanka on the 19th of December, 2012. Chemical analysis showed a relatively low density of the material, as well as a high content of carbon and organic substances. With the help of electron microscopy, scientists were able to detect round structures inside the meteorite fragments, about 1/10 of a millimeter in diameter, reminiscent of water bear eggs. Threads were observed on the surface of the structures, which could be the tentacles of these algae. As an argument in favor of the extraterrestrial origin of the structures, rather than contamination already on Earth, scientists cite their location inside the meteorite fragments and the low nitrogen content. And in 2018, a report by Russian specialists came out with the results of the study of microbes from cosmic dust that had settled on the outer side of the International Space Station Illuminator. Viable bacteria were detected, and to explain how they got to a height of 400 kilometers above the Earth, 2 versions were considered. The hypothetical mechanism of Earth's ionospheric lift in space as an alternative origin of these microbes. There is another side to the process. After all, space travelers could be not only tardigrades and water bears. Scientists seriously consider viruses as cosmic carriers of life. For example, there are studies suggesting that the so-called Cambrian explosion, a sudden increase in skeletal fauna biodiversity about 540 million years ago, was caused by the arrival of viral genes on Earth. In the fossil remains of that time, representatives of all types of animals existing today, including chordates, appeared over a short period. The coevolution analysis of retroviruses and their terrestrial and fish hosts showed that viruses appeared in the ocean 455 million years ago, at the beginning of the Paleozoic. According to astronomers, on the eve of the Cambrian explosion at the end of the Ediacaran period, the Earth was subjected to meteorite-comet bombardments associated with the passage of the solar system through a gigantic molecular cloud. This happens infrequently by cosmic standards, as the Sun, while revolving around the center of the galaxy, passes through many gigantic molecular clouds. This is a well-established scientific term. Just imagine, the entire galaxy, or even possibly, the entire group of galaxies, including ours, could constitute a single interconnected biosphere, not just a cosmic sector of space. As a result of this, on one hand, there are mass extinctions, and on the other hand, there is an active replenishment of the Earth's gene pool with cosmic genes, which are incorporated into terrestrial genomes and drive evolution further. And viruses are ideal candidates for such incorporation. They make up at least 8% of the human genome. This allows us to conclude that natural selection in humans and their ancestors occurred in partnership with hundreds of viruses. Their viability makes them practically invincible. In the process of evolution, some viral genes, embedded in the genomes of cellular life forms, began to perform important functions for the organism, such as the placenta. This red circle on the right allows both people and this little mouse to start life in the mother's womb. Perhaps this remarkable organ would never have appeared if our distant ancestors had not caught a very strange infection somewhere. The PEG10 gene necessary for the development of the placenta appears to have been acquired by ancient mammals from a mobile genetic element of a retrotransposon. A retrotransposon is something like a simplified virus, almost having lost its infectivity. That is, the ability to transmit from one host to another, but still capable of multiplying within the host's cells and integrating its copies into the host genome. They are usually transmitted only by inheritance from parents to children, but sometimes extremely rarely. They can still be infected with them, like viruses. This is not the only case of its kind. The retrotransposon leads its lineage ferment telomerase, which depends on the restoration of chromosome ends, shortening after each replication. Here's another example. One of the genes playing a key role in learning and memory, the gene. This virus-like gene is necessary for long-term storage of information in the brain. The protein behaves like a virus and serves as a platform for neuron interaction, as if it opens a window through which memories are solidified. And this window cannot be opened without this gene. A group of scientists led by Jason Shepard, a neurobiology lecturer from the University of Utah, who has devoted more than 15 years to the study of proteins, believes that the retrotransposon, a precursor of the retrovirus, entered the organism of mammals millions of years ago, which subsequently led to the development of the protein. A mouse with the gene turned off is incapable of learning and forming long-term memory. Having found cheese in the maze, she forgets the way to it the next day. But that's not all. Viruses are not only very successful at integrating into the genome, but also at cooperating, disguising themselves, and exchanging messages with each other. This discovery was made by scientists from the Weizmann Institute under the leadership of genetic microbiologist Rotem Sorek, who published the results of the research in the journal. The authors of the study suggested that one of the proteins serves as a means of communication between viruses, changing their behavior. After 2.5 years of experiments on bacteria of the genus Seno, they identified this substance. It turned out to be a viral protein released from the killed bacteriophages of bacteria. By the way, if you look at what bacteriophages look like and what they do, which are viruses that infect bacterial cells, the question of the extraterrestrial origin of these monstrous mechanisms, it seems, is not even worth it. So, the bacteriophage communication protein represents a viral peptide, which consists of 6 amino acids and is produced when the phage infects the bacteria, and they signal to other phages that the host is infected. When the number of signal peptides, and therefore infected cells, reaches a critical level, all viruses, as if on command, cease active action and go dormant. If it weren't for this deceptive maneuver, bacteria could organize a collective resistance or die completely, depriving the viruses of the opportunity to parasitize them further. Viruses lull the vigilance of their victims and give them time to recover. The peptide that helped them do this was called arbitrium, decision. Further research has shown that viruses are capable of making more complex decisions. They can sacrifice themselves during an attack on the cell's immune defense to ensure the success of the second or third wave of invasion. They are capable of moving from cell to cell in coordinated fashion, in transport bubbles, in vesicles, exchanging genetic material, helping each other mask from the immune system, cooperating with other strains to take advantage of their evolutionary advantages. There's a high probability that even these amazing examples are just the tip of the iceberg. And to study the social life of viruses, a new science has even emerged: sociovirology. It's not about viruses having consciousness, but social connections, language of communication, collective decisions, coordination of actions, mutual assistance, and planning can quite be considered as signs of intelligent life. Here's the most malicious example. Under the shell of the famous ball with suckers, there is a short ribonucleic acid molecule chain with 29,300 nucleotides. For comparison, our DNA has more than 3 billion such nucleotides. Quite a simple structure if you compare. But the virus doesn't need to be complex. The main thing is to become a key component of a complex system. Viruses can be compared to Somali pirates who captured a huge tanker on a tiny boat. They don't need the entire algorithm to control the captured cell. A short code that makes the entire cell's operating system work for it is enough. His code is perfectly optimized for this task. Inside the cell, the virus lives exactly as much as the system resources allow. In a simple system, it can divide and control exchange processes. In a complex one, like, for example, a human organism, it can activate additional options. For example, to reach such a level of information processing that borders on intelligent life. But why do viruses need this? To sacrifice themselves, help each other, and perfect their communication process. What is their goal if they are not living beings? As strange as it may seem, the answer is directly related to us. By and large, a virus is a gene. The primary task of any gene is to maximize its own replication, to spread in space and time. But in this sense, a virus is little different from our own genes, which are also obsessed, above all, with the preservation and replication of the information recorded in them. In fact, the similarities are even greater. Humans are a bit like viruses. The human body has accumulated about 98,000 retroviral elements, which once infected our ancestors. Now they make up 30 to 50 families, which are divided into almost 20 groups and subgroups. According to geneticists, the last retrovirus that managed to become part of our DNA infected the human population about 150,000 years ago. Then our ancestors survived a pandemic. What are relic viruses doing now? Some never show themselves, or rather, we don't know about these manifestations. Others work quite successfully, as we have found out. They protect the human embryo from infections, restore replications, chromosomes, participate in thinking and memory. But perhaps the mission of viruses is much more significant. It used to be thought that bacteria were only harmful, but as knowledge accumulated, it turned out that they are not only useful, but in many cases, vital. Just like that, perhaps soon they will stop demonizing viruses. Often they bring us disease and death. But what if the purpose of their existence is not the destruction of life, but directed evolution? After all, we exert evolutionary influence on each other, not just as environmental factors. Our cells directly participate in the assembly and modification of viral RNAs, and viruses directly interact with the genes of their hosts, introducing their genetic code into their cells. It turns out that a virus is one of the ways our genes communicate with the outside world. However, it's hard to get away from the thought that viruses, among other things, can arrive not only on the top of comets, but also in such a teacup. After all, not only bacteria, viruses, and organic molecules can be brought in. Almost ready-made organisms can be brought in as well. For example, a huge amount of evidence points to the extraterrestrial origin of octopuses, squids, and other higher cephalopod mollusks. An enlarged brain, a sophisticated nervous system, complex camera eyes, unusual jet bodies, and instantly changing shape and color. All these facts are very difficult to explain with standard sequential evolution. It's worth noting the fact that octopuses actively edit their ribonucleic acid. With the exceptional conservatism of DNA, spontaneous mutations in DNA occur in most animals, determining their further development. Meanwhile, squids and octopuses are capable of deliberately altering their ribonucleic acid to better adapt to their surrounding environment. Such modifications affect the production of proteins, which allows the organism to better adapt to existing conditions without resorting to actual mutations in the genome. As early as 2015, scientists discovered that a squid can edit up to 60% of its nervous system's ribonucleic acid, trying to better adapt to changing temperatures. And recently, an analysis of thousands of ribonucleic acid samples showed that similar capabilities are also present in octopuses and mollusk cuttlefish, which primarily inhabit warm seas near the coast. There are many theories of human origin to this day, but 3 of them are the most popular. These are the theory of evolution, creationism, and the extraterrestrial or cosmic version. Let's look at the facts that allow us to look differently at the generally accepted points of view about the origin of man. There is an aquatic theory, or the theory of the water monkey, also known as the aquatic ape hypothesis. Like the savanna theory, it is just a hypothesis, but nonetheless explains some aspects of human development. It claims that after leaving the jungles, our ancestors went to the sea, river, or lake, not the savanna. Swimming and diving. These are some of the characteristics of a human being associated with the aquatic theory. Modern humans can dive due to voluntary control of the breathing process. Moreover, humans have what is called a reflex to close the respiratory tract when submerged in water. This reflex automatically triggers when water reaches the face. The respiratory throat is not separated from the esophagus. Lower throat. A similar structure is only found in aquatic mammals, for example seals. It allows to control breathing, hold it, and dive. The vital necessity of the human body for iodine and sodium chloride salt found in abundance in sea products. The absence of iodine in consumed products leads to diseases of the thyroid gland. The opportunity for a full-fledged diet exclusively with the gifts of the sea, for example, Japanese cuisine. The presence of small webs between the fingers is found in about 7% of people. Some are born with webs between their toes. Humans have a web between the thumb and index finger, while primates do not. The presence of vernix in newborns is also characteristic of marine mammals, but not monkeys. The presence of a swimming reflex in a newborn, which is an atavism in modern humans. A large volume of fat tissue in the mammary glands is characteristic only for humans. This can be explained by the fact that milk had to retain heat in cold water. In monkeys, the mammary glands are small and without fatty tissue. All this is well and good, but perhaps Hydropithecus did not swim on this planet. Or it doesn't matter where he swam. The main thing is that he served as a donor of genetic material. A series of historical documents testify to the cosmic origin of man. In Sumerian mythology, the process of creating man is described in the most detail. Sources suggest that in the beginning, the world was ruled by gods, who were likely extraterrestrial beings resembling humans, according to modern interpretations. They came from the heavens to earth, the conquest of which required considerable effort. The god Enki proposed to create a man to shift the burden of labor onto him. One of the gods was sentenced to death. As stated in the source, his genetic material, flesh and blood, were mixed with clay. From the resulting material, the first man was created, similar to the gods, but being a slave to them. Therefore, the expression God's slave has a quite literal meaning. In other ancient texts, there is also often a mention of beings similar to humans, created by Enki and the mother goddess. And here's what's interesting. The results of the mitochondrial DNA analysis showed that all people are descendants of one woman who lived about 200,000 years ago, so-called mitochondrial Eve. The latest research by scientists suggests that there are 23 genes in the human body that are not found in other inhabitants of Earth. Pervtorin, or blue blood, created in the early 80s of the 20th century, is suitable for all people without exception. Possibly in ancient times this term blue blood was used in the literal sense, and this term was specifically related to representatives from other lands. Human blood, like most animals, is red due to the presence of iron ions in the respiratory pigment hemoglobin. But there are organisms that have hemocyanin based on copper instead of hemoglobin. The blood of such creatures is blue. These are some insects and mollusks. According to one version, on the planet where the gods came from, there is more copper than iron. And evolution there went in such a way that copper was used to transport gases and nutrients in the blood. The body temperature of 36 and 6/10 degrees Celsius is also not accidental, as at this temperature water has minimal heat capacity. Let's turn to the graph. To maintain the water temperature in the body at the mark of 36 and 6/10 degrees Celsius, the least amount of heat is needed than at other temperatures. This means less fuel in the form of food, less strain on cells and the body's systems as a whole. This temperature is low for the destruction of viruses, but there is a mechanism for raising the temperature to 40 degrees and above when many bacteria and viruses die. Man was not created for life in earthly conditions, as evidenced by his physiological inability. Specialists note a small number of such inconsistencies. Why, for example, did evolution give man constantly growing hair on his head to survive? But this is absurd. Imagine a child, an ancient man who, in the absence of scissors, has hair interfering with his vision, movement, escape. It's impossible to explain the constant and fairly rapid growth of hair on the head through evolution. After all, if there is some anomaly, like with this hairless one, then the fur is completely lost all over the body. For a woman to separate from her kin is also much more difficult than for most animals, and the period of helplessness of a newborn human is anomalously long compared to the same animal world. Back pain and other musculoskeletal disorders are associated with poor skeletal adaptation. In current planetary conditions, a human breaks extremities, falling from the height of his own growth, which is not observed in animals. A person can exist without clothes only in a very narrow temperature range of 20 to 26 degrees. The thermoregulation system is based on evaporation. That is, clothing is mandatory, and a full hair cover is not assumed. This is an unresolved question for evolutionists. What happened to the full hair cover? Magnetic storms significantly affect a person. The frequencies of Schumann resonances, or rather, changes in their parameters too. Breathing during long runs increases so much that the body clearly lacks oxygen for all exchange processes. The biological body was designed for a different ratio of gases in the atmosphere, as well as a different sleep and rest regime. The so-called circadian rhythm of a person is not equal to 24 hours. People who are in complete isolation for a long time, such as speleologists, usually switch to a longer daily cycle of 26 to 32 hours. Moreover, there is a known case of a 56-hour circadian rhythm. This mysterious feature gives reason to believe that a 24-hour day is not natural for the human body. If we were to shrink down to the nano level and take a journey inside a living cell, we would see motors, conveyors, assembly lines, and even walking robots. According to biologists, about 40 known molecular machines are functioning in a living cell. They carry loads along molecular rails, act as switches for chemical processes. Molecular machines produce energy to sustain life, contract our muscles, and build other molecular machines. For example, the proton adenosine triphosphate synthase is the smallest biological electric motor in nature, only 10 nanometers wide. With its help, living organisms produce adenosine triphosphate, which consists of adenosine accumulating a lot of energy. This conserved energy is needed to power a variety of biochemical reactions. Kinesin is a linear molecular motor that moves along the cell along the pathways of polymer threads. Like a porter, it carries all possible loads of mitochondria, lysosomes, using adenosine triphosphate molecules as fuel. Externally, kinesin looks like a toy man woven from thin ropes. At the same time, if one end of it uses adenosine triphosphate molecule energy while moving, the other releases a component for adenosine diphosphate energy formation. As a result, there is a continuous cycle of energy supply and consumption for useful work. Research has shown that kinesin can climb up the cell quite briskly with its filamentous legs. Taking a step of only 8 nanometers per second, it moves across gigantic cell distances of 800 nanometers. That is, it takes 100 steps per second. Try to imagine such speeds in the human world. walking along the microtubules, kinesin transports various loads within the cell. All this seems like a product of a civilization that knew how to create artificial forms of life. And recent research confirms that it is quite possible for a human to create artificial nanorobots that can live and work in the intracellular world in the future. Back in the early 90s, English scientists from the University of Sheffield made the first molecular machine, a synthetic device in which spatial displacement of molecules occurs. Rotaxane, an artificial substance in which a ring molecule is threaded onto a linear molecule, is used to create it. In 2010, a group of American bioengineers led by Milan Stojanovic and his colleagues created a molecular nanorobot capable of moving along DNA. During the experiment, the scientists were able to track how their nanorobot was able to independently make 50 steps and move 100 nanometers. The robot, externally resembling a spider, can autonomously execute several commands: go, turn, stop. And just 3 years later, English and Scottish bioengineers under the leadership of David Lee were able to create the world's first molecular nanobot machine capable of assembling peptides, short proteins. In nature, this task is performed by ribosomes, organelles found in our cells. Bioengineers took a molecule of rotaxane as the basis for their machine, and on its stern they were able to assemble a protein of a given property from separate amino acids. Thus, it can be assumed that if Earth scientists are capable of creating artificial life, then why is this inaccessible to other civilizations? So, discussions about the Creator have a different meaning from this perspective, right? But let's take a step back and ask the main question. Even if the single biosphere is not our tiny planet in the backwaters of the Milky Way, but a whole group of galaxies with billions and billions of living planets. So where is the source? Where is that star system from which everything started? And how exactly did everything there turn out so successfully that life began to spread on a cosmic scale? After all, at the moment, there are several main scientific theories about the origin of life, and about twice as many unscientific ones. This includes the primordial soup theory and the clay, not water, origin, where the optimal environment for the accumulation and complication of complex organic molecules is determined by clay minerals. This is due to the birth of life at the bottom of the ocean near black smokers, where the harmful radiation of the young sun could not reach. This includes protocells, the ribonucleic acid, world hypothesis, and even the surprising endosymbiosis. It was noticed at the end of the century that chloroplasts, the organelles of a plant cell responsible for photosynthesis, replicate separately from the cell itself. Soon a hypothesis emerged that chloroplasts are symbionts of photosynthesizing bacteria that were once swallowed by a host and have remained here forever. Later, the endosymbiosis hypothesis was expanded to include mitochondria, organelles that consume oxygen and provide energy to all our cells. To this day, this hypothesis has gained the status of a full-fledged, multiple times proven theory. It is enough to say that mitochondria and plastids have their own genome, more or less independent mechanisms of division from the cell, and their own protein synthesis system. In nature, other endosymbionts have been found that do not have billions of years of coevolution behind them and are at a less deep level of integration into the cell. For example, some amoebas do not have their own mitochondria, but they do have bacteria included inside that perform their role. There are hypotheses about the endosymbiotic origin of other organelles, including peroxisomes, plastids, and even the cell nucleus. We can examine all these versions in more detail in one of the upcoming series. If you find this interesting, let us know with a like and a comment, and set up notifications so you don't miss anything. That's all for today. Thanks for rating the video, and for the expert opinions from advanced evolutionary biologists in the comments. See you soon!